Hello everyone, this is MedBite here, the first video of the channel. I'm going to start off the channel by talking to you about what the different infectious agents are and giving you some background information on each one of them. So let's start off by talking about what an infectious agent actually is. An infectious agent, otherwise commonly referred to as a pathogen, refers to a microorganism or biological entity capable of causing disease in a host. The most prominent, or rather one of the most prominent infectious agents that I'm going to start off with are viruses. A virus is a microscopic infectious agent. Unlike other pathogens, viruses are unique because they lack ribosomes. And since ribosomes are required for protein synthesis, viruses themselves cannot actually produce any proteins. That's why they need to invade host cells in order to use the cell's ribosomes to synthesize proteins. So let's discuss the structure of a virus. A virus is essentially an infective genome, and it always consists of a genome and a capsid as seen in this diagram right here. The genome is made up either of RNA or DNA, but never both. And the function of the genome in a virus is to encode information needed to produce more viruses. The genome is protected by a protein coat known as a capsid. The capsid, or the main function of the capsid, is to protect the genome, but it also plays a part in enabling the virus to enter another host cell. In some viruses, but not all viruses, there is also an additional structure known as an envelope. So an envelope is essentially a membrane-like structure that surrounds the capsid of certain viruses. It is composed of lipids, fatty substances, and proteins. Again, I'd like to stress that the envelope is only present in certain viruses, like influenza and HIV viruses, but not all of them. Now, there's one important term I'd like to introduce to you here, and that is a nucleocapsid. The genome of a virus and the capsid of a virus are collectively referred to as the nucleocapsid. Now, there's another very important distinction I'd like to draw here and also introduce a new piece of terminology, and that is the difference between a virus and a virion. A virus refers to any biological entity which contains a nucleocapsid and an optional envelope. However, a virion refers to something slightly different. You see, a virus has a very complex life cycle, and at various stages of this life cycle, the virus is not infectious, i.e. they cannot infect a host cell. A virion refers to the virus during the life stages where it is competent to invade another host cell. A virion essentially refers to the fully formed and matured virus particle that is capable of infecting a host cell. Now, let me just quickly clarify this. If you have a virus which is unable to infect a host cell because it's just in a different stage of its life cycle, then you don't refer to that virus as a virion. There is another very important concept I'd like to talk about, and that is the concept of a subviral agent. A lot of textbooks don't talk about these, but I will, because I want everything to be complete for completeness' sake. Subviral agents refers to biological entities that are smaller and less complex than viruses, but still have the ability to cause infection. So that's why we call these infectious agents subviral, because they contain some of the components you would find in a normal virus, but they also lack some of the components that you would find in a normal virus. So they are subviral, they are inferior to a virus. The first subviral agent I'd like to start off with are prions. Prions are misfolded cellular proteins which influences the normal folding of other proteins and causes them to fold abnormally. These misfolded cellular proteins, aka prions, then aggregate together and they cause disease via various mechanisms. An example of a prion disease is Creutzfeldt-Jakob disease, where prions aggregate in the brain, causing destruction of important neurological tissue. That's Creutzfeldt-Jakob disease, abbreviated to CJD. Moving on, the second subviral agent I'd like to talk about are satellite viruses. Satellite viruses are similar to a virus, except that they cannot replicate independently, and they also require the services of a helper virus to infect a host cell. A typical normal virus can infect a host cell all by itself, and it can also replicate within the host cell by itself, whereas a satellite virus cannot replicate independently even inside the host cell, and it also requires a helper virus to infect a host cell. So that's why in these aspects, a satellite virus is inferior to your typical normal virus. Good. And the final subviral agent that I'd like to talk about are viroids. Viroids are small infectious subviral agents that consist of a single strand of RNA. They lack a protein coat, they lack envelopes, they lack proteins in general. All they contain is a single strand of RNA. Viroids only really cause disease in plants. Good, so we've discussed the different subviral agents. Just to quickly go through why each agent is inferior to a virus, prions only consist of proteins. They don't actually consist of any genomic material. They contain no DNA and RNA. So they are subviral. They are inferior to a virus. Your satellite virus is similar to a virus, but it's unable to replicate independently and infect a host cell independently. So again, it is inferior to a virus in that aspect. And your viroid only contains the genomic part of a virus. It lacks proteins altogether. So in that way, your viroid is inferior to a typical virus. Moving on. Let's talk about bacteria. Bacteria are single-celled prokaryotic microorganisms. I'm certain you'd have heard about bacteria in school or just in day-to-day conversations. Some of the key features of bacteria which you must be aware of are as follows. Bacteria lack membrane-bound organelles. Bacteria also lack nuclei. Instead of a nuclei, or a nucleus rather, they have a singular circular chromosome containing their genetic material. And this singular circular chromosome is known as chromosomal DNA. In this diagram, the chromosomal DNA is represented by this mass of squiggly blue lines. The chromosomal DNA is freely floating in the cytoplasm. It's not encased by any kind of membrane. It's completely free. Another important thing I'd like to point out here is, you can see on this diagram, nucleoid is pointing to the chromosomal DNA. But this is a distinction I'd like to draw here. The term nucleoid doesn't refer to the chromosomal DNA, but rather, nucleoid refers to the region of the cytoplasm which is occupied by the chromosomal DNA. So there is a slight difference, and it's very important to be aware of this. Bacteria are also classified according to two main properties: the shape of a singular bacterial cell and the result of a staining technique. Now, I will be discussing more about this in the future, but what I mean by this is, let's say that your bacterium has a spherical shape. It will be classed as a cocci, C-O-C-C-I. And if your bacterium produces a positive result in the Gram staining test, it will be classified as Gram-positive. So the bacterium will be labeled as a Gram-positive cocci. Again, I will be discussing more about this later, so don't worry too much about this for the moment. Just know the bacterium are classified according to the shape and staining techniques. A couple of important things that I'd further like to point out is plasmids. So you can see here that here is a plasmid represented by these red circles in the diagram. Plasmids are small circular DNA molecules, and these plasmids are separate from the chromosomal DNA. Plasmids contain extra genes for the bacterium. In fact, a lot of the antibiotic resistance genes that some bacteria contain are found contained within these plasmid structures. And finally, last but not least, prokaryotes contain 70S ribosomes. Now, in this diagram, the ribosomes are not included, but bacteria do contain them, and their ribosomes have a 70S size. S is basically a unit to measure ribosome size. This is in contrast to eukaryotes, which have 80S ribosomes. So it's an important fact to keep in mind. Parasites. Now, parasites, in my opinion, are not particularly well explained. So I want to make sure that we have a crystal clear understanding of what this infectious agent entails. Parasites refers to any organism which spends a portion of its life in direct contact with the host, causing harm but not death to the host, whilst itself surviving and reproducing. In layman's terms, what this means is that the parasite is in contact with us. However, this definition isn't quite complete. Now, it does cover a lot of what a parasite is, but if we were to go according to this definition alone, then in that way, viruses and bacteria could also be classed as parasitic, because they also do spend a portion of their life in direct contact with the host, and they can also cause harm to the host but not death, whilst themselves surviving and reproducing. So a little extra bit that I want to add to this definition is that parasites refer to eukaryotic organisms, excluding fungi. Good. So if you were to take both of these criteria and put them together, then you would have a concrete definition for a parasite. So now that we've discussed what a parasite is, let's talk about the different groups of parasites. You have three groups of parasites. You have protozoa. Protozoa refer to unicellular parasites, and protozoa are usually found within the human body. Next, you have helminths, also known as parasitic worms. Helminths are slightly different from their protozoan counterparts in various ways. Firstly, helminths are multicellular. Helminths also often, but not always, lodge themselves in the human's digestive tract. So what happens is that the helminth enters our body via various modes of transmission. It lodges itself in the digestive tract, and whenever I eat something, instead of my body being able to absorb the nutrients from the food that I consumed, the helminth, the parasitic worm, takes up those nutrients. So it itself is surviving, reproducing, and thriving at my expense. It's not letting my body absorb the nutrients that I need. That's just one of the ways in which helminths can affect us. They can also cause an immune response to be initiated, causing inflammation and more damage to the human parts, and also in various other modes, which I will be discussing later on. Now, helminths have a primitive nervous and excretory system, so they are a lot more developed than their protozoan counterparts. Now, let's talk about the classification of helminths. All helminths belong to one of two phylum. If you are not familiar with the concept of a phylum, then have a listen to the explanation that I'm going to give right now. If you are familiar, then you can skip the explanation via the timestamps and continue as normal. So, just deviating slightly, just to give you a better understanding, a phylum is a rank in the classification system. So, in classification, we need to have a very foolproof, a very airtight mode of classifying an organism. If we have an organism, for example, we need to determine which domain it belongs to, which kingdom it belongs to, which phylum, which class, which order, family, and etc., all the way down to the species. For example, let's take your local domestic cat. If we just call it a cat, that's very vague and ambiguous, and that's not very precise. Scientifically, it's not inaccurate, but it's not very precise. So, let's take the domestic cat. The domestic cat belongs to the Eukarya domain. It belongs to the Animalia kingdom. It belongs to the Chordata phylum. It belongs to the Mammalia class, the Carnivora order, the Felidae family, the Felis genus, and the Catus species, or the Felis catus species rather. So I've listed out each rank of the classification system for a domestic cat. So when I refer to a phylum, I refer to the Chordata. When I refer to the genus, I refer to the Felis, with regards to the domestic cat. Now, there's one little piece of nomenclature which I want to point out before we head back to parasites, and that is species. You can see over here that domain is one thing, kingdom is one thing, Animalia, phylum is one thing, Chordata, but species is two things. That's because when we refer to the species of an organism, we almost always refer to the genus followed by the species designation itself. So, for example, with regards to humans as well, we call humans Homo sapiens. Homo is a genus, the species is the sapiens bit. So, it's very important to understand that when you do refer to the species of an organism, that the genus is always capitalized. So, for example, here you can see that Felis begins with a capital F, but Catus begins with a lowercase c. That's very important. It's not a big detail, but it's a very important detail that you should certainly be mindful of when talking about taxonomic ranks. Anyways, moving back to parasites. As I mentioned previously, helminths can be divided into one of two phyla, and I've written the phylum below. They can either be divided into platyhelminthes, which are also known as flatworms, or nematodes, also known as roundworms. I cannot stress this enough. Any helminth that ever exists on this planet belongs to one of these two phylum. Of course, each phylum has further class designations, which have further family designations, and so on, but these two phyla are exclusive to helminths. Now, let's move on to the third group of parasites, which are known as ectoparasites. Ectoparasites are similar to helminths in that they're multicellular, but they're also different to helminths in that they live outside of the host. An example of a prominent ectoparasite are ticks. So here are the three main groups of parasites, some of the key features of each group, and moving on. And last but not least, we have fungi. Fungi are eukaryotic, and they can be divided into three major groups: yeast, filamentous fungi, and dimorphic fungi. So yeast, I'm sure you've definitely heard of. Yeast are single-celled, they reproduce via budding, and they're also the reason why your dough can rise. Filamentous fungi. So filamentous fungi are also referred to as filamentous mold, or even just mold. So when we talk about mold, we refer to filamentous fungi. So filamentous fungi, unlike yeast, are multicellular, and they have a rather complex structure. They contain hyphae and fruiting structures. I will talk more about these two structures later on, but for now it's important to understand that hyphae are thread-like structures which have important roles in nutrient digestion, and fruiting structures are important in the reproduction of filamentous fungi because fruiting structures produce spores which are released into the environment, and when these spores come into contact with the right conditions, they can germinate to form mold. And finally, our final group of fungi are dimorphic fungi. So dimorphic fungi are unique in that they can switch between their yeast form and filamentous fungi form. So they can actually exist in either form described above, and they have the ability to switch depending on the environment and certain other factors. So we've talked about the different groups of fungi. Another thing I'd like to point out here is that yeast, filamentous fungi, dimorphic fungi, these are not taxonomic ranks. These are not ranks in the classification system. These are just general groups that fungi can be grouped into. So we've talked about fungi, we've talked about all the other infectious agents, and I hope that this video was of some use to you. I do hope that this content helped you to gain a better understanding of the different infectious agents and how to classify some of them. I will be releasing some more videos that will give you a better understanding of the different types of infections caused by these infectious agents, and also a bit more information into the immune system and how that functions. Thank you very much for watching. Hope to see you soon.